So, good morning, everybody. Welcome after the summer break to another series of our um, institute seminar. Today, it's our pleasure to welcome Marina to our institute that she knows quite well. Marina obtained her degree in biology in 2005 at the University Complutense de Madrid. Following a graduate trainee period at the Severo Ochoa Center for Molecular Biology, she completed a master in molecular biology at the Autonomous University of Madrid. Then, Marina got a Comunidad de Madrid PhD fellowship and joined the group of José María Valpuesta at the National Center of Biotechnology, CNB. After obtaining her PhD degree in 2013, she moved to the laboratory of Dr. Dorian Bubeck in the Department of Life Science at Imperial College London. In 2017, she obtained a Juan de la Cierva Incorporación fellowship and joined Dr. Oscar Llorca group at the Margarita Salas Center for Biological Research, CIB. The following year, she moved to CENIO as a staff scientist, continuing her work in Dr. Oscar Llorca's group. Now, she has been awarded with Ramón y Cajal Fellowship, where she ranked very high in the Biociencias y Biotecnología area. And Marina will return to the CNB, where she plans to establish her own research group. And just to mention that she has very recently been awarded with the Premio José Tormo de Biología Estructural of the Spanish Biochemistry and Molecular Biology Society, as she already did also in 2022. So welcome, Marina. Thanks for coming.
Okay. Can you hear me well? Yeah. So yeah, I would like to start by thanking Javier and Esther for the invitation and the kind introduction, and for you to come here to hear about this work, which is the structural study of the gamma-tubulin ring complex, or gamma-TuRC, which is a multi-subunit complex that is involved in the de novo microtubule assembly. So you know that microtubules are important cellular elements in the cells that are not only involved in providing the support to the cells or define the shape and the size of the cells. They are also involved in key cellular processes such as cell division, cell signaling through the formation of the primary cilium, intracellular transport with the formation of the axons, or cell migration. But today I'm going to focus in this function, which is the cell division and the formation of this apparatus, which is the spindle, the mitotic spindle complex. So you know that during division, this mitotic spindle is formed and is mainly composed by microtubules. So in animal cells, the centrosome is the major microtubule organizing center, and during the cell division, this centrosome assists the nucleation of the microtubules that, in a very simplified model, they are going to reach the chromosomes in order to allow the sister chromatid segregation among the daughter cells. However, if something happens and this mitotic spindle is not correctly formed, we can have this kind of aberrant mitotic spindles, which is one of the main causes of the aneuploidy. An aneuploidy, you know, that is responsible for the chromosomal instability, which is one of the most important hallmarks in cancer. So I'm particularly interested in understanding how this mitotic spindle is assembled, and in particular how this assembly process is regulated both in space and time inside the cells. So as I have mentioned before, this mitotic spindle is mainly formed by the microtubules, and these microtubules are long polymers made up of alpha-beta tubulin heterodimers. These heterodimers polymerize in a very organized manner, defining the polarity of the microtubules, which is one of the main features of these structures. So thanks to this polymerization, we can distinguish two different ends in the microtubules. The minus end here, where the de novo assembly of the microtubules, or what is called the nucleation of the microtubules, starts, and then this plus end, where the microtubules elongates. And this differentiation is very important for the biology of this system. However, one of the main features of the microtubules is that these are very dynamic structures. So they can quickly switch between a growing and a shrinking phase upon the cell requirements. And in fact, this feature is very important for these microtubules to carry out with these cellular functions, such as the cell division, the cell migration of the intracellular transport. So as I mentioned, these dynamic natures of the microtubules, it's very important, but at the same time, for the formation of the mitotic spindle, it is a challenge. Because for this structure to be assembled, these three conditions have to be accomplished. So the microtubules, for the formation of the mitotic spindle, has to be nucleated in a specific spot, which is the centrosome in this case, and only in the centrosome. The assembly of this mitotic spindle has to be coordinated with the cell cycle. And what is also very important is that during the formation of this mitotic spindle, this minus end of the microtubules has to be stabilized to block the polymerization of the microtubules, at least during the length of this chromosome segregation phase. So in charge of the nucleation of the microtubules, like 20 years ago or more, it was discovered this multi-subunit complex, which is called this gamma-tubulin ring complex, or γ-TuRC. This complex is embedded into the pericentral material of the centrosome and is considered as the major microtubule nucleator in cells. And this γ-TuRC complex is responsible to assist to accomplish these three conditions. So this γ-TuRC is specifically recruited into the centrosome, and only when it's bound to this pericentral material of the centrosome is active. So this is a way that the cells prevent the ectopic nucleation of the microtubules. So it's only active when it's at the centrosome. The gamma-tubulin or the γ-TuRC recruitment factors, the concentration of this factor at the pericentral material is also cell cycle dependent. And not only that, this γ-TuRC has been proposed to, once it nucleates the microtubules, it's proposed that it remains at the minus end of the microtubules, acting as a stabilator for them. In fact, if you inhibit some of the components of this γ-TuRC, you will see later the name, but these proteins called GCPs in general, you can see that the formation of this mitotic spindle is completely affected, is aberrant. So what we wanted to understand here was the microtubule nucleation mechanism that is behind the function of this γ-TuRC complex. But in order to understand the mechanism of this γ-TuRC, we need to understand how this complex is activated. We need to have a look to the structure of the complex and also the assembly process. And in the future, but I won't talk about it today, I would like to use all this information to try to block the activity of the γ-TuRC. So starting from the beginning, let's have a look to the γ-TuRC structure and the assembly mechanism. So again, this is the γ-TuRC that is the pericentral material of the centrosome, and it was proposed more or less the composition of this protein, but it wasn't. There was no much structural information about it. All the pioneers' work, the structural work made on this γ-TuRC, came from the yeast homologs. So in yeast, it was solved the structure of what it was called the gamma-tubulin small complex, which is the γ-TuRC subunit. And in yeast, it's composed by these two proteins called GCP2 and GCP3. Each of them are holding one monomer of the gamma-tubulin. And what was proposed, but not observed at the time, is that this γ-TuRC unit might homo-oligomerize to form this kind of ring-like structures. So here we will have one singleton of the gamma-tubulin monomer, and this singleton of the tubulin monomer might act as a template for the microtubules to be nucleated. But again, this was a hypothesis. It wasn't proved. So with this information, our collaborators set up an in vitro reconstitution system for the γ-TuRC. So all the γ-TuRC components were co-expressed in insect cells infected with a recombinant baculovirus. And then by affinity purification, we managed to obtain this nice sample that we image at the microscope. So here you can see some of these particles highlighted in green, and these are some of the average images. So the first thing that we observe is that, yeah, this is a ring. So these gamma TASC complex are almost oligomerizing as it was predicted. So this is the final structure that we get by cryo-EM, and we, thanks to achieve a high resolution, we managed to identify all the components of this structure. So you can see here that we have this, it was called the gamma-tubulin small complex, which is homolog to the GIS gamma-TuRC, composed by GCP2 and GCP3, but also that, in fact, this gamma-tubulin small complex is repeated up to 5 times in the complex. But we also found some eukaryotic specific gamma-TuRC components. So beside these GCP2 and GCP3, we found GCP4, GCP5, and GCP6 forming what it was called two gamma-TASC-like complexes. Okay, and this was only found in eukaryotic. So we solved the structure of the human gamma-TARC, but it was also solved the structure of the Xenopus complex, and it has exactly the same composition. But the most striking feature that we had here was the presence of this active monomer at the lumen of the complex. So it was it was a really surprise for us, because we didn't include this actin in the expression cassette of our baculovirus. So this actin, in our case, came from the host. It was also solved the structure of this complex, purified from native sources, and in every case from human and Xenopus, it was always found one molecule of the actin at the lumen of the complex, whose function was completely unknown. So yeah, so that was something quite fun to play with. So this is the general architecture of the gamma-tubulin components, these GCP proteins. So they are composed by one C-terminal domain called GRIP2, which is involved in the interactions with the gamma-tubulin. Another GRIP domain here that is involved in the lateral interactions with the gamma-TASC complexes. And also, we found this N-terminal extension. So it was observed that in every GCP protein but GCP4, it was found this helical domain whose structure was completely unknown, structure and function was completely unknown. So in this work, what we observed is that they are involved in interactions with gamma-TARC factors. In this case, they were called MOSAR1 and MOSAR2, and this factor were proposed to be involved in the assembly, but the function wasn't observed. So thanks to our structure, we realized that this complex of the N-terminal extensions with these MOSAR proteins, these modules were involved in the assembly of the gamma-TARC. So for instance, the N-terminal extension of GCP2 were involved in the stabilization of the intra-gamma-TASC interaction, while the N-terminal extensions of GCP3 and GCP6, also in complex with MOSAR proteins, form this kind of luminal extra density here, or a structure that was called the luminal bridge. And this luminal bridge, you can see that is contacting different gamma-TASC units, so it seems to be involved in the inter-gamma-TASC interaction. So in general, these N-terminal extensions contribute to the assembly and stabilization of the complex. Another important observation that we did here, it was in fact something that a problem that we encountered when we were trying to co-express all the proteins together, is that this complex, the gamma-TARC, needs these two proteins to be fully assembled. So these proteins, RABEL1 and RABEL2, are AAA ATPases that forms an heterohexamer that are involved in the assembly and maturation of many and diverse macromolecular complexes, including the mTOR or the polymerase II. So these two proteins, we observed that in the absence of these two proteins, we couldn't get this fully assembled complex. In fact, if we inhibit one of these proteins, RABBL1, and it has been observed that when you inhibit RABBL1, also you see a decrease in the levels of RABBL2 because they are mostly working together. So when we inhibit this RABBL1, we observe these central duplication defects in the cells. We also observe these mitotic spindle defects and chromosome congregation defects. And specifically, when you don't have RABBL1, we observed that in our purification, we couldn't obtain this final complex, but intermediate or gamma-TuRC structures. So yeah, so here I show you an image of the experiment that we did to try to analyze the implication of this RABBL complex in the assembly of the gamma-TuRC. So if we co-express all the components of the RABBL, of the gamma-TuRC without the RABBL, that were the only structure that I could observe in the microscope, so gamma-TuRC complexes or some intermediate complexes. Only in the presence of the RABBL complex we can have the fully assembled gamma-TuRC, and in this case I couldn't observe any isolated gamma-TuRC or intermediate of the complex. I really tried to get a structure of this gamma-TuRC complex with the RABBL complex, but I didn't manage to get an interaction, a stable interaction between them. So yeah, so the conclusion here is that, okay, the RABs are helping the assembly of this complex, but they are not part of the, they are not an integral part of the gamma-TuRC, which is the case for other gamma-TuRC clients, but not in this case. So what I did was to try to use these gamma-TuRC complexes and mix them with the RABBL complex in an in vitro controlled conditions and add a little bit of cross-linker to see if I was able to see the complex. And yeah, I can kind of see this transient complex having this cross-linking, but the structure that I get was a very, very low resolution. So yeah, so the RABBL complex is important, but we don't know exactly the role of this RABBL complex in the assembly of the gamma-TuRC. That's something that I would like to continue studying. Okay, so now that we have had a look at the assembly of the complex, we can see what the structure can tell us about the mechanism of action of this gamma-TuRC. And just for you to remember, the hypothesis is that this gamma-TuRC serves as a template for the microtubules to be nucleated. So yeah, so this is again the structure, and something that I intentionally omitted before is that this structure is asymmetric. So in cells, most of the microtubules are composed by 13 protofilaments, and if you count the number of spokes of this structure, you can see that we have 14 spokes. In fact, if we superpose one turn of the microtubule on top of our structure, you can see that while half of the complex follow the geometry of the microtubule, the other half present this helical deviation, what prevents this complex to be a template for the microtubules. And accordingly to this observation, in all the in vitro microtubule nucleation assays, we obtain a very, very low nucleation activity compared what was expected to happen in cells. So the conclusion here is that this complex, and not only our complex, all the structures that were published the same year, analyzing the same subject, present this low nucleation activity. So the conclusion is, okay, we have this purified complex, but this purified complex is not active. So the next obvious question is, okay, how this complex become active? And we start to have this idea of that maybe this gamma-TuRC is not working as a microtubule template, because so far it is not a template. So let's start trying to understand this activation mechanism, and for that we collaborated with Jens Luders and Fabian Zimmermann from the IRB in Barcelona. So they were really trying to find a list of candidates for the activation of the gamma-TuRC, and they found the CN1 domain of this centrosomal protein, CDK5-RAP2. That was one that it was in the first position in this ranking list of the candidates for the activation of the gamma-TuRC, because it was previously observed that this is a gamma-TuRC recruitment factor, and it was known that if the gamma-TuRC is not recruited into the centrosome, it is not active. it was suggested that it might stimulate the nucleation activity of the gamma-TuRC, and it was observed that there is a direct interaction between the CN1 domain and the gamma-TuRC, because it was used to, or it was co-purified with one of these complexes purified from natural sources in another of these structural studies. So what our collaborators did was to clone this complex, purify and express it, and they proved that it has to act as a dimer. So they built this dimeric construct for this domain. So once they had the CN1 domain, they just added into this microtubule nucleation assay, and as you can see here, it really boosts the nucleation activity of the microtubules. It was increased almost 5-fold times. So, okay, so having this result, the next obvious question was, okay, the CN1 is promoting the closure of the gamma-TuRC ring to become symmetric. So what we did was to add this CN1 into our sample, purify the complex, have a look to this complex by Cryo-EM and solve this structure. And you can see here that we have exactly the same components that we have before in this asymmetric and open gamma-TuRC structure. It was observed that this CN1 dimer interacts specifically with GCP2, that if you remember, it is there are up to 5 copies of the GCP2 protein. So in fact, yeah, it's stoichiometrically bound to the complex. So in every single CN1 binding region in the complex, we found this extra density here in orange, and in all of them, we managed to build the atomic model of this CN1 dimer. Okay, so this activator is bound in the complex, and it is bound stoichiometrically, so it's completely saturated. But if we compare the structure, the overall structure or conformation of this activated gamma-TuRC with our previous non-activated gamma-TuRC, you can see that there are no significant differences. So there are small changes here, but the overall conformation is the same. So seems to be still asymmetric, or seems to be in this open conformation. However, when I was processing this data, something that I realized compared to the open gamma-TuRC is that, or the previous gamma-TuRC, is that this subunit here were much more heterogeneous. It was more complicated for me to get a stable density to model and to map and model. So I confirmed this observation by using this Occupy software. And you can see here in this local scale that in this kind of reddish colors, you can see that there is a significantly increased heterogeneity compared to the rest of the components of the gamma-TuRC. So I say, okay, so this complex is roughly the same, but there is this increased heterogeneity. So I took all this data set, and then I tried to analyze this heterogeneity using a deep learning-based technique, which is the method, which is Cryo-Dragon. And using this method, I managed to obtain a range of different conformations around the ring seam that seems to represent the different closing stage of this gamma-TuRC. Here I can show you a morph between the more open in CS1 and the more close CS3 conformations that I found. So it's not exactly the same as the open one. In fact, I took the data set of the non-active gamma-TuRC, repeat the same experiment with Cryo-Dragon, and in this case I saw some heterogeneity around, but you can see that the conformation around the ring seam is more or less the same. So next I say, okay, I was really focused on getting these close rings. So I say, okay, I'll take the particles from the more close conformation, I extract them, and then I'll process them separately in order to reconstruct the more close conformation that I can get from this data set, and that's what I did. I get the structure, and then I build a model, which is here shown in blue. And you can see that, as you can see, is still not a perfect template for the microtubules here in orange, but it's something in between the structure of the inactive gamma-TuRC and the structure of the microtubules. we did some measurements, and it was quite evident, especially if we measure the helical pitch. So here in blue, this more close conformation of the active gamma-TuRC seems to be more similar to what is expected in the microtubule. In this activated gamma-TuRC structure, another important feature that we observed was related to this luminal actin. So as I mentioned before, it was quite a surprise to find this actin molecule here, and it was proved that it's an integral part of the gamma-TuRC. And it was also observed a couple of years ago that this actin is not involved in the gamma-TuRC assembly. So if you delete it, you can have a fully closed gamma-TuRC structure, but it's important in its function. So if you don't have actin, the gamma-TuRC does not nucleate microtubules at all. So in this case, I also observed during the processing that the occupancy of the density assigned for the actin wasn't as strong as I found it in the inactive or non-active gamma-TuRC. So I used the same software, Occupy, and you can see here that compared with other structures, such as this luminal bridge, you can see that this one, the occupancy percentage is significantly lower. So I say, okay, if this is an important difference, I'm going to classify the particles based only on the occupancy of this active molecule, and that's what I did. And by doing so, I obtained two different classes. So the main one is this class 1 containing the actin, but 30% of the particles completely lack this active molecule in the lumen of the complex. So based on the position that this actin has here, that is quite close to the seam of the ring, it was hypothesized at that time that maybe it is affecting or is inhibiting somehow the closure of the ring. So maybe we need to get rid of the actin. That was also observed in this experiment, that it was, at the beginning, it was intending to understand the mechanism for the CN1 activator. So as I have explained you before, all the gamma-TuRC components are co-expressed in vivo, so the assembly of this complex takes place in vivo. I don't have any in vitro assembly model to study it. So, but I wanted to see if the CN1 was somehow assisting the assembly of the gamma-TuRC. So what I did was to take only the gamma-TuRC complex, which is the only the gamma tubulin small complex that is homolog to yeast. That's the only one that I can purify in isolation without the other gamma-TuRC-like complexes. So I take this complex and then I add it into the sample, this CN1 dimer. And you can see here that, in fact, this CN1 dimer is promoting the homo-oligomerization of this gamma-TuRC. You can see that using the same concentration of the gamma-TuRC unit, in the presence of CN1, the number of rings is significantly higher. And I wanted to get the structure of these homo oligomers of this gamma-TuRC complex, but it was quite complicated. But I did observe some extra density also bound to GCP2 that at that time I wasn't sure, because I didn't have the other structure. I wasn't sure if it belonged to CN1 or not. So what I did was to have as a control the same gamma-TuRC homo-oligomers in the absence of CN1. But you can see that this homo-oligomerization without CN1 is very unspecific. So I added a little bit of cross-linker. And with this cross-linker, I get a very nice structure of this gamma-TuRC homo-oligomer. And as you can see here, it doesn't contain this extra density. So that was another proof that this density corresponds to CN1 before. That was done before I could model the CN1 domain. So this is the structure that I obtained, and you can see here that this structure is in the closed conformation. So there is this was another proof that these gamma-TuRC-like components, these eukaryotic specific GCP proteins, are involved in having this asymmetry in the human gamma-TuRC. But yeah, but this experiment also served me to have a look to this actin that was not present here in this complex. So if I fit the atomic structure or the atomic model of these spokes from 2 to 4 in complex with the actin, you can see that the position of this actin is, it has this steric class with this subunit here. So yeah, so this is an indicator, that was an indication for me that the active might be affecting the closure of the ring, because it seems to be in the middle when the ring is closed. So yeah, so we can kind of summarize up to here from this non-active gamma-TuRC structure, which is asymmetric, it's open here, and it contains this luminal actin preventing the closure of the ring. We can activate this complex by adding this CN1 domain, and although this complex remains in this open conformation, we can see that this actin molecule becomes mobile, and also the whole complex around the ring seam explore different conformations. And among those conformations, I found one that it was significantly closer or close to the final position that it was expected. However, here we did this observation that the activation of the gamma-TuRC is not equivalent to the ring closure. So this brings us to the next question. Maybe this gamma-TuRC is not a microtubule template. So it was a very important question to be answered in the field, because all the previous models were based in this idea of the microtubule template. So in order to answer this question, we contact Thomas Surrey and Claudia Brito from the CRG in Barcelona. They set up a very nice in vitro microtubule nucleation reaction. They use this biotinylated gamma-TuRC structure and then bound it to the streptavidin in a cover slip, and then they add these alpha-beta tubulin heterodimers. So this was exactly the same as we did before. So what was expected is that some of these gamma-TuRC structures might nucleate microtubules, but in a very inefficient way. So the final microtubules nucleated, the number were very, very low. So what they did was to substitute these wild-type tubulin heterodimers by some mutant tubulin heterodimers. So they introduced some, in fact, this mutant was previously introduced or published. So they introduced this mutation in the alpha monomer that hydrolyzes the GTP much more slower. I haven't explained that, but during the tubulin polymerization, the hydrolysis of the GTP at the alpha tubulin monomer is important. So yeah, so at the end, by using this mutant, they managed to increase the number of microtubules nucleated. So, but the problem here is that, okay, now the gamma-TuRC is active, it can nucleate microtubules, but those microtubules were very, very long, because this is a very quick reaction. So for me in the microscope, to have these long microtubules is a problem. So the next step was to add this DARPin molecule, which is an engineered protein that specifically binds to the beta tubulin subunits of the tubulin heterodimers, and it blocks the addition of extra alpha beta tubulins on top. So the idea here was to get very short microtubules. So we took this sample, we moved into Cryo-EM, and we spent a lot of time trying to get the optimal conditions, balancing between the number of gamma-TuRC with the microtubule nucleated and the length of the microtubules, because these kind of images that were quite spectacular at the beginning, they are a nightmare to process the gamma-TuRC and to remove the density of the microtubule. But we found some nice conditions, and then we managed to get, for the first time, the cryo-EM structure of the gamma-TuRC in complex with a nascent microtubule. But not only that, we managed also we observed that there was a high level of heterogeneity, and we managed to reconstruct different steps of this gamma-TuRC along the process of the nucleation of the microtubules, what allow us to analyze not only the initial closing step of the gamma-TuRC, but also the final steps when the microtubule start to elongate. But let's come back to the structure of this gamma-TuRC. So you can see here that compared to the previous structure, the composition of the complex was conserved. So we have the same number of GCP proteins bound to this gamma tubulin, and on top of them, we observe one turn, at least one turn, of the alpha-beta tubulin heterodimers on top of it. So yeah, again, we still have these 14 spokes in the structure, but now this complex is nucleating 13 protofilament microtubules. So this complex can nucleate these 13 protofilament microtubules by placing the position 14 right on top of the first one, in a way that the addition of alpha-beta tubulin heterodimers on top of the first spoke is blocked. So we have these 13 protofilament microtubules. Here you can see a comparison of the conformation of this open gamma TuRC and then this fully closed gamma TuRC. You can see here the gap that, I mean, there is a gap here in the lateral position of the last spoke, but there is also a difference that is not well represented here in the helical pitch. So it's much lower. While this one, yeah, it is acting as a template for these 13 protofilaments to be nucleated. Okay, so we wanted to understand how was this closing mechanism for the gamma TuRC. So here you can see represented in cartoon the structure of the fully closed gamma TuRC structure superposed with the structure of the open gamma TuRC, the non-activated gamma TuRC in surface. So you can see at the beginning we thought that because when you superpose this turn of the microtubule, you can see that half of the ring was mimicking the geometry of the microtubule, but not the other half. So maybe we thought that there was kind of a hinge for this complex to be closed, but that's not the case. You can see that is more like a concerted conformational change of all the GCP proteins of the complex. So you can see that these GCP's proteins undergoes a conformational change from a more bent conformation to a more straight conformation. And what it does at the end is it has this kind of iris-like movement that allow this complex to maintain the same number of GCP proteins, but in a more constrained space. We also observe that the actin was different. So this is the structure of the activated gamma TuRC with the actin at the lumen of the complex, and in this fully closed conformation, the actin is not there. We don't know if the actin is released or the actin gets disorganized, but it's clear that the actin wasn't there. But you can see that these two differences that I found, they seems to be more like accommodating the closure of the ring more than promoting the closure of the ring. So what is really driving this conformational change? We found a new gamma TuRC structure element that we call it the latch here, that is acting, this additional density is acting like a bridge between the last spoke in position 14 with the gamma tubulin in the first position. And not only that, it is also contacting the gamma tubulin monomer of the first heterodimer added into the second position. This structure was described for the first time here, but it wasn't observed for the first time in this work. It was observed in the non-active gamma TuRC structure, but here it was quite heterogeneous, so you can see that the density, the complete density wasn't well defined. And it was also observed in our active gamma TuRC structure. In fact, here it was more organized and it was already contacting this gamma tubulin, but we didn't have the tubulin heterodimer, so it was not finding the third or the second anchoring point. I really tried to get high resolution of this structure, but it was impossible. And the reason is that this structure is very heterogeneous. So you can see that we found kind of different stages for the latch formation during the microtubule nucleation. From this initial latch structure, the fully formed latch, and then I didn't manage to see any latch kind of structure once the microtubules start to elongate. And maybe it makes sense that I don't see the latch, because once the microtubules start to elongate, the tubulin lateral interactions start to be formed, and of course the longitudinal interactions. So the contacts of the microtubule lattice maybe are enough to keep the gamma-TuRC close, and this latch structure is no longer required. But we are still working on it. So as a summary, from this activated gamma-TuRC structure in the presence of this CN1, we can add these alpha-beta 2 heterodimers. So the actin is released, the latch is formed, and this complex becomes a template for the microtubule nucleation. And that's it. I would like just to thank all the people and that has been involved in this work. Of course, Oscar, my boss, and the CNIO, Rafa Leiro from the Genome Integrity and Structural Biology group, also the CNIO that helped me a lot in the modeling of the first asymmetric gamma-TuRC structure. That was quite complicated for me, and in fact, he called it the Capilla Sixtina. I would like also to thank Jaska and Joanne from the CNIO electron microscopy unit, and my collaborators, Jens Luders and Fabian Zimmermann from the IRB, working with this active gamma-TuRC, Thomas Sarri and Claudia Brito working with this gamma-TuRC in complex with the microtubule. And the data that was acquired at Brem with the help of David, and at Alba with the help of Pablo. And that's it. I would like to thank you for your attention.
Thank you. Thank you, Marina, for this beautiful talk. Now let's have some discussion, some questions from the audience. Anybody?
Beautiful work. Congratulations.
Thank you.
So have you been able to identify what is the latch? So were you able to do some mass spec or something? Do you have any idea?
So yeah, we did mass spec, and we didn't find anything extra from the gamma-TuRC compared to the non-active gamma-TuRC, for instance. So yeah, I think I know what it is, but I cannot prove it because I don't have the resolution. So I have shown you some structures of these N-terminal extensions of complex with this Mozart protein. So some of them are forming these luminal bridge, some of them are involved in the with other functions in the outer part of the complex. But if you count all this extra density from N-terminal extensions, there are 3 that are missed. In particular, one of these gamma-TuRC asymmetric, these gamma-TuRC-like units, the GCP5, whose N-terminal extension is completely unlocalized, let's say. So it has been also observed by cross-link and mass spec that it interacts with these Mozart proteins. So I did an AlphaFold prediction of this extension with one of these proteins, and then I fit it, and it fit perfectly. So that's what I suspect is happening. So now the next obvious step here was to add everything together, and that's what we did. So now we have gamma-TuRC, CN1, and the microtubule. And what I have now is the fully closed conformation in a much more stable, or in a final step, let's say. Instead of having all these different steps in the nucleation, I only have the fully closed with the elongated microtubules. And in this structure, I can see the latch, and it's much more stable. So my hope is that once I focus the refinement in this region, find the identity, or I can prove the identity. But yeah.
I have a second question, and it is about the role of actin in inhibiting the closure. So that would this be a way to inhibit, to target, would it be a way, an interface to modulate closure and therefore inhibition of this complex to be assembled?
Yeah, so the actin is complicated, and in fact we still don't know what's happening. So when you activate the complex, a certain percentage of the protein doesn't have the actin. Then when you have this kind of early nucleation steps, the actin is not there. It's not there meaning that I cannot see it. I don't know if it's there, but disorganized, so I cannot refine it. So something that we did was to add DNase1, for instance, that is known to be a interactor of the actin with a very, very high affinity. So we start the nucleation activity or the nucleation reaction, add this DNase one in order to capture all the actin that might be released from the microtubules, and then polymerize the microtubules, purify the gamma TuRC, and have a look to the composition. The actin is still there, so it's not released. In fact, in this new structure, I can see density at the same position once the microtubules is nucleated and elongated. So the actin is not released. We know that. But it gets destabilized somehow, or the conformation has changed somehow. Because if you, in this fully closed structure, if you fit the position of the actin based on the interaction regions with all the GCPs, it cannot fit, but it's still there. So yeah, we don't know what's making this actin to be destabilized to allow the ring closure. Yeah, we still don't know. We are working on it. That's very, very interesting, because it's not related to the assembly. That has been proved. You can have exactly the same complex, the same structure without actin, but it cannot nucleate microtubules. So yeah, it's very interesting, because one might think that you don't have actin, the ring is closed, you can activate more microtubules, but that's not the case. So maybe it's doing something else, not only inhibiting the ring closure, it's doing something else with other elements of the gamma TuRC. We don't know. Yeah, might be. Yeah, that's very interesting. Thanks.
More questions? So, beautiful talk. I was kind of like, to try to follow up the previous question, I was wondering, because this actin, the role of this actin monomer there. So with this Cryo-Dragon software is very interesting. Would you think that with the time you would be able to kind of follow this allosterism in the way that the actin monomer is being released, so that the gamma TuRC can go like full power? I don't know.
Yeah. So for me, the first step is that now I have this final structure, because the actin is dead, but the conformation is different. I think if I can analyze the structure now of the actin and see how the interactions with all the gamma TuRC components has been modified, maybe it gives me some ideas about this mechanism. So I have to confess that when I was starting with this Cryo-Dragon, I just wanted to have a look to general changes, because the obsession in the field was, okay, how or this to get the structure of the closed gamma TuRC. So I just focus on getting the closed structure. But now with Cryo-Dragon, you can apply masks. So having the structure, you can focus the kind of classification in some regions of the complex, keeping the rest of the complex being the same. So yeah, I can try to apply. This complex is huge, and the actin is very small. So every time that I apply a mask into the actin, everything gets a little bit weird, let's say. So the controls has to be very, very well thought. But yeah, that's a very good idea, just to mask and analyze the heterogeneity to see different conformations of the actin, or different interactions of the actin with other elements. Yeah, I'll do it. Thanks.
All right, so no more questions?
You know, to produce this complex, so you infected viruses with different viruses, because did you know the stoichiometry at the beginning?
No, we didn't know.
You didn't, so you just individually infected with different viruses coming from the different proteins?
No.
Or you do produce a cassette and fingers crossed?
Yeah.
So
in fact, what we did at the beginning was to build this recombinant baculovirus, only one recombinant backmeat with all the gamma TuRC components. It didn't work. Then we added these Mozart proteins, Mozart 1 and Mozart 2, the ones that are involved in the interactions with the N-terminal extensions. Kind of work. We start to see gamma TuRC complexes and some intermediate structures, but we need to add this Rabiel complex, and that was added in using another virus. So yeah, so now what we have is 2 viruses, and then we mix them, infect cells, and then purify using tags in this GCP5, 4, or 6. Because if you use the other ones, you can get a mixture of everything. But we are using 2 viruses now. But all the gamma TAR components, let's say the integral components of the complex, in 1 virus. Only one copy of each. Yeah. Yeah. So yeah, I don't know. We try at the beginning. We try, like, each time we clone something, just try and see what happens. But only having all of them, the yield of the purification was good. If not, it was complicated.
Okay, so let's thank Marina
Okay,
again
thank
for
you.
this beautiful talk at the visit. And we would like to give you a little present from the Institute as a souvenir and a reminder of your visit. Thank you.
Okay, thank you very much. Thanks.